So this week, students are going to be exploring several different sort of content areas as well as skill areas. One of the major things that I've often found my students don't realize is that those French fries or those potatoes are made up of thousands of cells, if not millions of cells, lots and lots of cells. So they don't think about this piece of potato is made up of lots of cells, and those cells are going to behave in the ways that we've been learning about in our class based on the semi-permeable plasma membrane and what can and cannot cross it. So you may want to sort of remind them gently that those little French fry pieces they're working with are made up of lots and lots of cells. And they should have prepared for lab by reading part of the textbook. If they've covered some of this in their lectures, that's great. They also have a mini lecture on tonicity that reminds them of some of these ideas. So they'll be using potatoes as models for learning about cells. They also are going to be learning about tonicity, hypotonic, hypertonic, isotonic solutions, and how that affects water relations in cells. So they'll be understanding that. They'll be thinking about the idea that while we're in sucrose solutions, sugars are large molecules that will not cross that membrane on their own through diffusion, but water can cross that membrane through osmosis. So that's the kind of thing they should be looking at. They're going to use what they have learned either before coming into the lab, by doing some reading, by sort of digging into the lab, and they're going to be using the data that they collect to predict the mass of a cell given relative tonicity of a solution. So at the end, they should be able to, if you give them sort of a quiz on it, they should be able to predict what will happen to the water balance in the cell and how we predict that and what that really means. Another thing they're going to be working on is following a protocol. Now, this is early in the semester usually. You really want to try to get them to read these protocols and understand these protocols and ask you questions about them before they dig in or as they're going. It can be very tempting to just tell them the answer to things, but I highly recommend that if they ask you a question that's in that lab protocol, don't belittle them, because a lot of students can have very I'll say poor reading comprehension skills, or especially if they're nervous, they maybe can't understand it because of that. Just kind of go over there and say, okay, let's look at the protocol together. And what that will do is train them to, hey, look at the protocol first, see if it's there. And hopefully by the end of the semester, they'll be really, really great at that. And that's one of our goals as well. This lab is also going to get them gathering data that they're then going to analyze. These will be unique data. It's different every time we teach it. It's not hugely different, but they'll have little differences caused by the actual individual who's doing the measuring, who's doing the weighing, who's doing the cutting. They can be different based on the structure of the potato. Was it a Wisley potato? Was there something wrong with the potato? There's lots of things to think about that makes these data actually unique. And they're also going to be learning about measurement and experimental error, which is a really important way to get them sort of understanding how science works and thinking about their techniques and their lab mates' techniques. So major tips and tricks are in the write-up. And I already mentioned, remind them that these are cells. These potatoes can be of any variety. We tend to use russets because they're cheap. You want a good medium-sized potato, not potato that you can sometimes see in the baking potato bags. And you want a healthy potato. So you want it to be nice and firm. If you wouldn't eat it, do not use it. So if it's wrinkled up or it's squishy, it's going to be not working quite the way we would expect it to. So a dry potato will suck up water no matter what solution you put it in. So we don't want them to be wrinkled or shriveled or squishy. It's a good idea to use a French fry cutter. Now, when we started doing this lab, we went cheap and we got the cheap plastic ones that for your kitchen would probably be fine. But we ended up going to a kitchen supply store and getting an actual kitchen, like professional kitchen potato slicer to make the fries. And I think that in the end, that was the cheaper way to go because we didn't have to replace it every semester. So either you can cut the potatoes using the French fry cutter, or you can have the students do it depending on your comfort level. One tip I would add is put some damp paper towels under the potato cutter so that when the slices come out of it, they come right onto a damp set of paper towels, and then cover them with damp paper towels as well. So you do not want to have these potatoes get dried out. It does say in the protocol the students should blot them, but they just kind of want to blot them, right? They don't want them to be totally dry. Take a damp paper towel and just kind of blot off any dripping water. Really important to remind the students to compare the potato before and after to itself. Sometimes I use the example of the TV show The Biggest Loser, which I don't know if that's still around, but you wouldn't want to compare potato A to potato 33 or something like that. You want to compare this potato to itself before and after to see if it gained or lost mass. So while the potatoes are in the sucrose solutions and you have kind of two options of tubes you might want to use, I really like to use the 50 ml screw cap tubes because they're easy. You put the potatoes and the solution in there and it's sealed and it's easy to do. You can also use large glass test tubes, and that's in the prep list given to you in these instructor materials. If you use the test tubes without a top, you're going to have the students use something like parafilm to seal the tubes. And almost none of them, if any of them, have used parafilm before. It's incredibly uncommon to have that happen. So you might need to show them how to use parafilm, which is fun in itself. Once they get the potatoes in, they're going to be doing a measurement activity, and there will be a little primer on that in this area of the instructor materials as well later. So if you need a little primer on what that means, there will be a short video on how to do that as well. Now, the data this week will be collected, and then they're going to learn how to do graphing next week in another lab, and then they will have that assignment due. So they get sort of 2 weeks to work on this assignment.